So my voice is okay? Can you hear? Yeah, great. Thank you. So thank you for the invitation and the introduction, and thank you all for showing up. So today I'm going to talk about my research, multi-dimensional associations among genotype, environment, and the phenotype. So you may notice that I put a symbol here, symbol, right? So nurture versus nature. Since 16th century, when the nurture and nature, these two terms came into use, and there are many people actually, this have been spent ample time for discussing which is more important. It's our human behavior determined by the genetics nature, or determined by the environment nurture. So this is an age-long debate. So I switched the order, nurture versus nature. That indicates I will emphasize or stress on the environment in my talk. So before I start, I would like to give a special thanks to Dr. Jianmin Yu, Dr. Xianran Li, and Adam. So we four. So we involved in a lot of wonderful projects, and this talk cover a lot of projects I'm working on, and we've been, doing a lot of exciting research that's very enjoyable. And also I thank the former graduate students and our current graduate students for their support and the suggestions on most of my projects here. And so I appreciate the help from the technicians in your lab, and this is the US collaborators and the international collaborators. And our funding is from our department and the Plant Science Institute, USDA, NSF, and Kansas State. So this is the first experiment. It's very early, actually, started very early. This is the twin, just a twin. They have exactly the same DNA, but they show different height. So because the DNA is the same, we fix that term. Then the difference of their height must due to the environmental difference, because the nutrition of their food is different. One is balanced nutrition food, one is per diet. And the other experiment show these 7 varieties. They were grown in the same garden, so the environment is the same, but the height, we see there's variance of plant height. Then this various plant height is determined by the genotypic difference. These two experiments deliver the information to us. First, environment is associated with phenotype. The second, genotype is associated with phenotype. So before I introduce you this this is a long-established experiment. It's very helpful. But to make my explanation clear, I would like to clarify on the definitions. The first, genotype, it is the genetic makeup of an individual organism. So for a particular trait, it is a site of gene on our DNA which is responsible for that trait. And the environment is the sum of all the surroundings of a living organism. So for a specific environment, you can think about it is weather, temperature, nutrition, or biotic factors. That's the environment. And the phenotype is very easy. It's just anything you can observe, you can count, you can measure, right? So this is our phenotype. Then this experiment, this is a very beautiful mountain, have 3 elevations. The low elevation, medium elevation, and the high elevation. So then there are 7 Achillea plants, but they have another name called yarrow. So these 7 varieties, we see this is the parental parents. The plants was cut into 3 sections, and each of the section, because there is called asexual reproduction. So the cuttings of the plant, they can grow into a whole plant if you put the soil there. So then that means this parent was cut into 3 sections. The first section planted in the low elevation, the second is the medium elevation, and the third is the high elevation. So that means if plants in the vertical position, they are identical genotypes. And the plants on the horizontal position, that means they are unique genotypes. So just to observe this result, we see the plant height are different, flowering time are different, a lot of different phenotypes, right? Then we think about this the plant, they grew very high in the high elevation and the low elevation, but they failed to thrive on the medium elevation. We think about maybe the medium elevation does not allow this plant to grow. Actually, it turns out that's wrong, because you see this parent, it grew very well. So what does this experiment deliver information to us? The phenotype of the genotype, of each genotype, depends on their environment. The phenotype of each environment depends on their genotype. So then, until now, we can comprehend or we can understand the phenotype is partitioned into two components. One is genotype, one is the environment. They also have interactions. Then the major components can be further dissected into different pieces. So the genotype can be further dissected into genes on different chromosomes. And the environment can be further dissected into environmental factors. For the plant, it may be the day length, temperature, wind, humidity, all of these pieces, right? So this part of work is what we are working on right now. So we know which gene responds to that trait. We know which environmental factor responds to that trait. But then the next question come into our mind, how much? We want to know how much contributions from genotype, how much contribution from environment. For specifically, we want to know how much one gene can contribute to the phenotypic variation, how much of one environmental factors contribute to the phenotypic variation, right? So then when we talk about quantification, the genetic model, the statistical model come into play. The ANOVA table actually is from Fisher, right? So this part of work is contributed by the Fisher and the establishment of this quantitative genetics. The Fisher, he used this ANOVA table can tell us to partition the phenotypic variance into the genetic variance, environmental variance, and G/E variance and errors. So then this is the Fisher told us. But the Finley-Vixen model, they told us how much of the G/E can be regressed. That means additive part, right? And then Amy model, GG model, the other model, they told us how much of the major component from the G/E contribute to the phenotypic variations. So this whole slide is introducing for the quantification. In the context of big data, we have big data every day. We generate a lot of data, multiple dimensional data. But this multiple dimensional data can be from either phenotype, or genotype, or environment, because the phenomics is to enable the development and adoption of high throughput phenotyping, right? I know a lot of professors in your lab is doing the phenomics. And also the genomics is to characterize the genome. What is the structure? What is the function of that genome? And the third is related to the meteorology, right? Meteorology is a study of the atmosphere that is focused on the weather precise and weather forecasting. So they can generate a lot of data. It brings us unprecedented challenges. So there's so many data, how can we use that data? But it also brings us opportunities to study the associations between them. Then genomic selection and the genome-wide association studies is two current methods to dissect the novel genes by using these two information to dissect genes, or use all the available markers to predict the phenotype. So these are two the current methods. However, we didn't use the environmental information here. Then my study is how do we incorporate the environment into genomic selection and the GWAS framework, help us to explain this phenotype. So genomic prediction is a method by using all the markers on the genome to predict our phenotypes. So we use the marker information to predict the phenotypes such that breeders, they can make selection without planting all the available lines in the field or without testing them in multiple environments, right? So then there are two methods for genomic prediction. One is the mixed model, one is the neural network. So I'm mainly working on this method. And one of our graduate students, James, he is working on that. I can see we are in good relationship, but we are competing with each other. Sometimes he said, oh, I'm better than you. Sometimes he said, oh, I just better than him. So in terms of genomic prediction, everyone can get the accuracy, right? Sometimes the accuracy is high, low, but how high is high? How low is low? Then I'm thinking about, we have a lot of discussions on if the prediction accuracy is 0.5, is that good enough for breeders to make selection? So how do we explain this 0.5? So I think this table can help us interpret a little bit. So the phenotypic variance is 100%, right? This is 100%. And then by going down to the prediction accuracy, it reduces a lot of information. Our prediction accuracy maybe is low to medium. 0.5 is here, right? So then we lost a lot of information here. Give you an example. So from the phenotypic variance to the broad sense heritability, what part is removed? The environment, G/E part is removed. We only consider genetics there, right? And also, from this genomic heritability to the prediction, it reduced, right? It is caused by the inconsistent of this LD status in the training and in the validation population. The LD status is different. It's caused prediction accuracy cannot catch this part. Then my scientific question is coming. How do we improve the prediction accuracy? Can we incorporate this environment in the prediction model in the first place? We conducted a very large-scale multi-environment trials. You see, it's multi-species, multi-location, multi-year, and multi-trait. So there is a lot of multiple here. So the species include oat, maize, wheat, and rice, and cotton. And the locations is global, right? And also the population is diversity panel. The phenotype we collected are mainly the morphological traits, flowering time, green yield, and some nutrition traits, some chemical traits. So a lot of traits here. The first, I did a partitioning. Which one contributed to the phenotypic variation? I partitioned the phenotypic variation into 4 parts. First is environment. Second is genotype. Third is G/E, and the residual is gray. So I think a lot of people, they look at this figure, they have a lot of questions, right? I have the same doubt. So why does the G/E account for only a small part of phenotypic variation? So does that mean G/E is not important? Or does that mean G/E can be ignored? So then this is our question. I will explain this topic later, but this is the question always in my mind. So the second question is to use the same data set. I do genomic prediction, the traditional genomic prediction. I only use genomic information, does not incorporate environment. Only genomic prediction, genomics in the model, I calculate the prediction accuracy. You see, for some trials, I just so disappointed, right? So the prediction accuracy is so low. How do we improve genomic prediction accuracy is my second question. Third, I incorporate environment information to the data set and do genomic prediction again. So we call cross-environment genomic prediction. You see, the accuracy increased a lot. So this is, we are so happy about our method, so I want to promote to everyone. Please try that. The third question is how to conduct a cross-environment prediction accuracy. The first question, is G/E important or not? So let's first look at how environment acting on this genotype. Environment is so powerful because environment changes the leaf architecture. For the same genotype, the leaf architecture is exceptionally different, right? And then for the butterfly, I mean, most of you may doubt, are they from the same genotype? Yes, it is. It is because environment changes their shape, changes their color, right? So then, let's look at the G/E. When people introduce G/E to you, a lot of studies introduce G/E is important. He used this figure, reaction arm. So just the genotype react to the environment phenotype, and the environment variable show this phenotype. This type of G by E is called crossover because there is a crossover, or we call rank change G by E. So from this reaction norm, G by E is so important. Then it's not very often we look at this type of reaction norm. This is based on two genotypes. This is based on the population level. So from the population level, what can you tell? How many G by E contributed to this reaction arm? See? Here, the very significant G by E is only a small part of the reaction arm on the population level. Then, actually, I can tell you the truth. This figure is from Fisher's potato experimental design. He showed the table, and I plot them to see how many G by E. And Fisher's conclusion is that G by E is not significant from error. So he said G by E can be eliminated from statistical studies. Then, because Fisher raised his opinion on how to partition the phenotypic variation to genetics, to environment, and he also said G by E is not significant from error. Then Hogben wrote mail to Fisher. Hogben, he said he is doing his experiment. He wrote email to Fisher. He said, if the environment contribution is 10%, and I uniform the environment, does that mean the phenotypic variation reduced 10%? And Fisher wrote back. He said, if the environment and genotype, they are independent, the model is additive. That works. And Hogben is confused by Fisher's answer. He confused. He cannot figure out this question, his answer. And then he wrote a very long rebuttal. He said, you see, I raised all my flies in the 16 Celsius. The variation is 5,000. And then I did a second experiment. I raised all of my flies on the 25 Celsius. The variance is 600. He asked Fisher. So which variance takes the priority in estimation of the genetic variance? And Fisher said, I see your point. You are concerning the nonlinear part. And Fisher does not understand the nonlinear. No, Hogben does not understand the nonlinear part, right? So then they wrote back and forth. So later on, they criticize each other in their publications or in the mails. So we look at their research. It's not just to see how these two science giants, they argue with each other, right? We want to learn something from their mails. So then we summarize that. Oh, let me see. So the Fisher's G by E is considered called biometric G by E. And Hogben's G by E is called developmental G by E. Unless I can tell the conclusion is it's very hard to make a comparison because the problem is different. Because Fisher, he wants to see how much, he want to do the variance partition. He wants to know how much contribute to the phenotypic variation, right? It's a statistic. But Hogben, he is individual development. He just look at the wild type and the mutant, right? Just the wild type and the mutant, if they have a crossover, G by E is so significant. And the Fisher is working on the population, right? And the methodology is different because the biometric G by E is we collect the data, collect a lot of data, and we analyze, use different models. Then this is our conclusion. But Hogben is, let's create an environment. Let's create, the temperature is extremely low, the temperature is extremely high. Let these two groups of flies, just they fly everywhere. And let's observe their behavior. So it's this method called intervention because they create the environment for them. So then there's a lot of difference for them. We did the experiment on both. So the first experiment we did is there is rice biparental populations. We conducted this experiment in 9 environments actually, and across 6 locations and 2 or 3 years. So on the left, this is the reaction norm on the population level. On the right, This is a reaction norm on the individual level. So these 4 panels represent 4 genes, and the 2 colors means 2 alleles. You can see from A and B, there is a G by E here. This is scale change, and especially HD2, it's rank change. If you look at only 2 alleles, they are very significant G by E because they cross over. But if you look at the population, how much of the G by E contributed to the phenotypic variation? Only 9%. This is another experiment. It's a sorghum biparental population. We already published this result. So it's the same. This population grew in 7 to, right now it's already 11 environments. This is a population, this is reaction norm on the population level. This is reaction norm on the individual level. How much of this G by E contribute? That's high, 20%. Then the first question or topic, I gave a conclusion said, biometric and developmental G by E focus on the population level and the individual level, respectively. Reaction norms on population level include multi-types of individual level of G by E. G by E is important, even though it explains a relatively small proportion of variance. So let's take a break. This is a cartoon to show why are you standing on the chair? And these people said, it's for perspective. Don't disturb me. I'm seeing the problem from different point of view. Because our study is individual level and population level from different perspectives, the result is different, right? So the second question, how to improve genomic prediction? In the context of modern plant breeding, we do plant breeding, right? So we have a lot of germplasms. We have a lot of candidates, lines, hybrids. Then we proposed a strategy for the genomic prediction or selection. We have the overall populations and the sequence because genotyping is getting cheaper and cheaper, right? But phenotyping them in the multiple environments is still challenging. So then we said, can we just genotyping all the available lines, get the genomic data for these available lines? And then we only use the genomic information to select a training population. This training population is do the phenotyping and to predict the other remaining lines. So this is a routine. Everyone is doing that. But we proposed the training population shouldn't be random. The training population should be representative. It must be very small but informative, can predict very well. So my question going here, assume this is our germplasm pool. How do we recommend a training set for the breeders to use? Then they can improve their prediction accuracy. I'm so happy by showing this figure is because this question is not only I face. A lot of scientists in different areas, they face the same question. It's called representative subset selection. So this problem has been faced by the social network, their scientists, and also it is the issue in the biology. It is also the issue in politics, and also this is also social network. So then, how do we select a subset training population from this complex network? First, I should select these two objects, right? Because they are bigger. That means they can connect the others very well. They have very high prestige. And the second, these two cannot be together. They should diverse, represent their own communities. So this is an example to show in our area. I said we should genotype in all of the lines first. What can we get? It is a marker matrix. We get a marker matrix. First, we transform them into a dissimilarity matrix. It's also called a genomic relationship matrix, or you can call that kinship. Then we transform this kinship to a network. The network is showing here. Then we do some magic here to make them dots, some show up, some does not show up. So then these 3 dots, I say they represent the structure of this network. So what is the magic here? It's called ranking algorithm. So I don't know. I don't know how many people they maybe this is already started, but I can see this is very easy. It's very easy because you just throw a bomb into this network, let them run. Random walk, just a random walk. You can go any direction, random walk. And then after they converge, some colors show deep color, some show blue color. That means I don't select that. And the pink color, I select them. So then there are many random walk methods. You can do random walk. You can do reinforced random walk. I mean, this slide is very complicated, but if you don't look at this slide, just look at me. Because here, if this left panel is I can do the random walk, right? So I'm in a community. I have 2 neighbors. So my name is called B73, and one of my neighbor is called Mo17, and one of my neighbor is called PH207. And then I visit my neighbors. I have 50% visit Mo73 and 50% visit PH207. And any random walk, I have 50%. So finally, to the end, they ask me to select which one can represent your community. It's hard to make a decision because Mo73 is my good friend, 50%. PH207 is also my good friend. So this is the left panel. But what is the magic about this right panel? It is, so I have 50% to visit Mo17, 50% to visit PH207. But the first time I visit Mo17, and then this visit just in my memory, I remember that. Next time when I do the random walk, I said, oh, I remember last time I visited Mo17. And then this time I also visit Mo17. So the probability or transition probability from here is increased 50 to 51. And the third time increased to 53. Then the PH207 decreased. The transition probability to Mo17 increased. To the end, they asked me to select a representative to represent our community. I said, definitely Mo17, he's my best friend. So then this is just to explain this. We can do random walk, but if you do some reinforced random walk, you can increase the diversity of your network. So we explore multiple data mining methods. The method I explained here is graphical network. We can also do the clustering analysis. It's unsupervised machine learning methods, right? Clustering methods, graphical network, and also genetic design methods. We apply these 3 methods into 3 data sets. One is maize hybrids, wheat hybrids, wheat hybrids and rice hybrids. Let's look at the result. So comparing with the random walk is a pink. And the 3 methods we said is called data mining methods. And in terms of prediction accuracy, you see, all of our data mining methods are better than the random sample, right? It's the same size, but its prediction accuracy increased. Compared to the other methods called the PV mean and the CDV mean, these 2 methods are from the mixed linear model, and considering the reliability or the prediction error to select, it's purposely selected the training set. So compared with these 2 classical methods, our data mining methods still generally outperformed. Then my second story, data mining tools and techniques can be leveraged in genetic studies and breeding strategies. My second conclusion, genomics enables breeders to identify superior genotypes from a huge number of candidates. So it's time to take a break. This is a cartoon to show. So every day, when the kids come back home, the parents' first question is, what did you learn from today's class? And the kids said, today we learned sequential patterns in data mining and several new clustering algorithms. And the parents said, reminded me to never ask again him what I learned in school. So we learn from the next generations. There are data mining methods we can use, right? Then this is the third question. how to conduct a cross-environmental genomic prediction. This is the reaction norm, the rice biparental populations. Flowering time, this is our phenotype, and the environment. This environment is the city name combined with the year. So this is different environment. And then this is the flowering time, show up and down, up and down. And one day, Dr. Yu gave me a task. He said, can you predict the new environment, FU09? Where is this FU09? So without access to the weather stations, I don't know where is this environment. I cannot put here, 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 here. So I cannot do prediction. Sorry, Dr. Yu. I need to quit my job because I cannot finish that. And then the next scenario. So it's the same task to do prediction for the new environment. But he said, I tell you the flowering time is responding to the temperature. And I can tell you the training set, the different, we already have the training environment. Can you do prediction on this almost 20 Celsius? This temperature, what is the prediction? What is your predicted flowering time? I said, that's easy to do. Because for the new environment, the temperature is here. I draw a line here. Done. It's done. It's very easy to accomplish. The next year, I planted in. I not only predict this, I predict a lot. I just overqualify my job, right? I predict a lot. And then I do predicted accuracy, 0.97. Oh, I'm so happy. I finished that. Prediction accuracy is so high. So I asked Dr. Yu, can you raise my salary? I don't need to quit my job, right? So from no to yes, from you almost need to quit your job to you can possibly raise your salary, there is only one step. It's you must know some information, just one step. You know the plants, they are reacting to this temperature. So how do we explore this temperature? How do we know how plants, which environment factors the plants reacting on? We proposed, these two plots show how do we find that. We explore the day length, we explore the temperature, and we do the exhaustive search and informative search. So then we figure out the temperature within 9 to 50 days is the best one. Plant can strongly reacting to them. But for this rice population, it's not responding to the day length. It's because all the 9 environment, they are in the day length conditions, long day conditions. We can see here they squeeze together. But in sorghum study, we said, okay, it's reacting to the photothermal time. It is a product of day length and temperature. And also the window changed. So this is just a very simple figure to say we can do the cross-environment prediction. But here I see genomics. We can do the cross-environment genomics prediction. The first, we can do prediction by using genomic information or kinship. It's because the new lines, new genotype, can be linked to the tested genotype by genomics. And the environment can be linked by the environmental factors. Then there's there's unlimited. So we can predict everything. You see, you can predict here, 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 even here. New genotype in new environment. So how do we, the challenge task is coming here. The new environment, new lines, how do we do this kind of prediction? So first, we predict the slope and the intercept with kinship. For some line, we're using the slope and the intercept to make prediction. Second, we predict the environmental index using temperature here. Third, we use joint regression analysis to predict the phenotype. So it's pretty easy. You just plus this, because this A and B is predicted here. And environmental index is predicted here. You plug in this regression model, you can get the predicted flowering time. This is the prediction accuracy. R is equal to 88. It's pretty high, right? So then I make a conclusion. Identifying key environmental factor is crucial in cross-environmental genomic prediction. Second, accuracy and on-target values can be achieved by applying our prediction methods. Now it's time to take a break. So everyone have different expressions on this cartoon, right? Move one step further. It's very easy because you just move one step further. But it's very challenging because this one step further is very hard to move. Because we need to think about our method. We have thousands of discussions in our lab. And we need to write the program, the package that people to use. We need to figure out all the detailed information. It's very challenging. So this is my ending slides, almost the ending slides. I have 3 ending slides. First, go deep. I talk about the genotype, environment, and phenotype. This time, I'm going to talk about the gene, temperature, and the flowering time. So gene is dissected, temperature is identified. They react to the flowering time. We can see here, I detected 4 major flowering time genes, HD1, HD2, 5, and 6. So for HD1, in the low temperature, it can delay flowering time almost 15 days. But in the high temperature, it's inactivate, right? HD2 is very interesting. HD2 in the low temperature, they delay flowering time. But in the high temperature, it promotes flowering time. So we can see these genes, the genetic effect, the additive effect, responding to the temperature. This is another reaction arm figure. And B, we do the QTL mapping for the intercept and the slope. We see HD1, 5, 6 are major genes responding to the genotypic effect intercept. And for the slope, it means sensitive to that environment, right? So HD1 and HD2, they are very sensitive to the environment, to the temperature. It's inconsistent with our result here, right? So thanks for the high coverage sequence data on the 2 parents, we can figure out what is the functional site, what is the functional polymorphism on that, which kind of amino acid change, and what is the structure change. We figure out the polymorphism on these 4 genes. And last, we know these 4 genes are in the major branch of the flowering time pathway under the long day conditions here. That's why I said we need to go deep, go the gene level to study that. The second, we need to go abroad because that 4 genes was discovered in the biparental population. And also it's discovered for responding to the photothermal time. No, the photo period. And we discovered that there is also responding to the temperature. So go abroad. We look for these 4 genes in the 3,000 rice genomes project. There are almost more than 3,000 rice cultivated accessions. They sequence them, very high coverage, sequence them. And then we find out what is the functional site of these 4 genes in that 3,000 accessions and do the haplotype analysis. We see the haplotypes, the functional site can separate them into indica and japonica. For all of these genes, all of these 4 genes, they are very important. And also I did this, I projected the different haplotype combinations. You see here, HD1, different color are different haplotypes. The different haplotype combinations projected into their origin countries. Here, original country. We can see here, this haplotype, yellow-green, this haplotype adapt mainly to the north part. The others adapted to the south part. So only 4 genes and different haplotypes, they adapted to their local environment, right? And also, I can see is the adaptation is significant or not. I do the FST test. So the combined FST value 0.42 is significantly adapted to their local environment. So this is my slide. Go far, go deep, go broad. Last is go far. Dr. Yu said this figure is the best figure I drew during my many years postdoc career. So he said, you should say something on that. So honestly, I still don't know what should I say on this figure. But I want to say something else. So one day my son is playing the toys, and I just check the news. I check what is the significant science questions in the future. And they have questions on how to figure out the core of the Earth moving, and how to figure out the black hole. And how do we figure out how the genome or DNA, they really want to tell us what. So this is all of the science questions. I'm so shocked. I just shaking. Oh, the science is so powerful. So my son is asking me, Mom, why are you shaking? I said, the science is so powerful. And then my son said, oh, then don't do experiment. So then why I want to say that is because, so we cannot imagine 100 years ago, there is a scientist, he's doing this pea experiment, Mendel's pea experiment. He cannot imagine after 100 years, we can figure out all these genes, where are they, where they are, and their functions, and how do they respond to the temperature, how do they respond to the day length, right? Maybe these scientists, if they know that, they will also shaking, right? Just the science is so powerful. And then I go back to this figure. When I drew this figure, I said, this is, so two brains, left brain and right brain. This is genetics, this is environment. So the first stage, Mendel discovered this genetic law, right? And then this double helix tell us, we just guess, they have gene controlling this trait, they have gene. They have some genetic factors. And then this double helix, Watson and Crick, he tell us, okay, gene is on this DNA, the DNA will wear something. And then, so right now we know these genes, they connected to each other. The omnigenic model tell us, okay, all the basic genes, they're working together, and it looks like our brain, different neurons, networks, right? And the right brain is the weather, because there is the same, there's no at that stage, there's no large-scale experiment. So right now we can access the weather stations, the climate change. We can use the equation to represent that. So it's more mimic to our two brains, left brain and the right brain. So then I almost finished my presentation. Go far. We are on the way. Thank you. Oh.
So I have two questions. The first one is about your the multi-species
experiment.
I know a table.
Uh-huh.
How much? So you show that the wheat flowering time data. There's only worry the genetic effect is almost neglected. They're only worrying you. Yeah. They're only.
Here, right?
Yeah, the genetic effect is almost neglected.
Yes.
And a lot of them are residual and your environmental factors.
Environment, yes.
In your prediction, you can still capture substantial amount of predictability.
Here, right?
Go back. Yeah. You can see you can still have about
Yes.
0.2 prediction accuracy.
Yes.
Yeah, you can still have 0.2 prediction accuracy. So how do you explain that?
So the within environmental genomic prediction is determined not by the environment, right? But it's determined by the red part divided by the green part. So that means we ignore this environment and consider this is 1%, and how much the red part take a part of this red plus this gray. Think about the heritability we calculate, we ignore the environment out, right? So that means here we still have heritability there. Then we can do prediction.
I mean your prediction accuracy.
Yeah, we do. Yes.
0.2 prediction accuracy.
Yeah, I know. But when I, yeah.
It looks like it's not in proportion with genotypic variance compared to the environmental variance, right?
Yeah, we don't consider environmental variance here. We just consider how much the genotype take a part of all this genetic plus this G by E plus this arrow.
And my second question is about your second project. It looks like your main goal is to identify properly identify the relationship between the different genotypes, right?
Yes.
So you use mixed linear model.
Yes.
So I'm curious. Did you only use a kinship matrix, or you also tried other kernel like the Gaussian kernels chapter non-additive feedback?
So this is the hybrids, right?
No, you can go to your histogram. Your bubble.
Here, right?
Yeah.
Yes. We use the non-additive part. When we do this subset selection, we use the kinship, right? But the kinship, actually we want to use the covariance matrix, variance covariance matrix, right? So the kinship. We use the kinship plus the variance, the additive variance, plus the kinship generated by the dominance times their dominance variance, and get them together to get a dissimilarity matrix and to do the selection. And also when we do prediction, we not only consider additive, we also consider dominance. Yeah, so we consider that.
Excellent talk. So my question is about the cross-environment genomic predictions. So how would you find the boundary for cross-environment genomic predictions? Say, if you want to predict the phenotype of a rice by parental population in a location of in Arizona. So is that possible, or is that meaningful, or what's your expectation of the prediction accuracy for that?
What is the expected prediction accuracy? It's depending on your training set. If your training, you do the regression is very well, right? And your horizontal, the environment factors is within our region. It's not outside of that, because we know the prediction. It should be within your boundary. The accuracy is good, right? So then think about maybe this is your horizontal here, and then we plug in to calculate that. So it's depending on your training population. Davis? Yeah.
So this seems fairly good in retrospect. And as you said, machine learning can only tell us things about what has happened in the past.
Yes.
Okay. So with climate change, we are told that we should expect things that we've never seen before.
Yes.
Can we start to use this framework to start predicting for environments that we've not seen? In other words, can we adapt soybeans to Manitoba?
I understand. So yes, when we discover all of these methods, when we discover how to our methods, we use the data we collected. We already have the weather information. We already have the flowering time. We already have everything. And then we discover that. We just assume we don't know. That's cross-validation, right? We assume we don't know. But here, the amazing part is we do that. You see, it's actual value. We predict within 5 days with observed data within 5 days. So we have a term called on-target in-season prediction. So in-season means we don't predict next year right now. We predict until next year between, we just planted there because we have a window here. So it's 9 to 50 days. So we use this window. It's just the in-season here, and then we make prediction. But like you said, if I want to predict next year what happened, we must use historical weather data. So it's just the other area. If the weather or climate people, they can predict very accurate of next year's weather. That's great, actually. But right now, if we cannot reach that, we're just getting close to that day it happens.
As follow-up, have you looked at the predictions that the climate modelers are forecasting for next year? How good are they 1 year out, is what I'm asking.
So our result, it is this, in the sorghum paper, we predict ELWA 15 and 16, right? That is,
Say that again?
we predict the plants planted in the ELWA is 15, 16, 17. We do the empirical validation, right?
Okay. That's not the question. The question is, have you talked to climate modelers and find out how good are they right now at predicting next year?
We haven't talked that.
Okay,
but
so they are doing this right now.
Yes. Okay.
And they have been doing this since 2005.
Uh-huh.
And so you should go to those data
Yeah.
resources and say,
Uh-huh.
okay, this is what you predict for Iowa. They can do a region like Iowa.
Uh-huh.
They can do Ames, Iowa, but they can do Iowa.
Yes.
In fact, they can do different quarters of Iowa.
Yes,
And
we.
say, how
Mhm.
good are you
Mhm.
last year at predicting this year?
Okay.
And how
Yeah.
good are you at predicting? And they'll tell us what next year.
Yeah, that's very good suggestion.
So-
Yeah.
And they predict temperature,
Mhm.
rainfall, and they do it on a quarterly basis.
Yeah, that's great. That's the cross-discipline. We can talk with them and we can work together for the next year's
We can
prediction.
Right now,
Yeah.
project when these will flower next year.
Yeah.
Put them in the ground next year and find out how good.
Okay.
Yeah.
That's great. Thank you. Hi.
I have another potential opportunity question.
Uh-huh.
So your environment seems to really focus on climate.
Uh-huh.
Is climate explaining enough of the environmental contribution, or is there room for soil?
Yeah. So to answer your question, yes, definitely it is. It has room for soil, but it depends on your experiment. So we have a talk. Well, we just talked to another professor, Sotiers, and he explained because our experiment is large scale. You see that, and he said, okay, distance and temperature actually can explain 90% of this phenotypic variation. He talked about this with us. That's for large scale environment. But for your data, it's the soil. If we do some experiments just for different types of soil, and then we can definitely say the soil contribute to that. So, yes.
All right, we're going to end on that question. Please help me thank our speaker one more time.